Welcome to EPG Pathshala. Hello students, I am Sunil Khare, Professor of Biochemistry in the Department of Chemistry, IIT Delhi. Today I am going to teach you about the nitrogen cycle and nitrogen fixation. In today's lecture, we will see how nitrogen is mobilized in the environment via nitrogen cycle. We'll also study about the different modes in which nitrogen is fixed, with special emphasis on biological nitrogen fixation and its importance in the ecosystem. Further, we'll explore the underlying biochemistry and genetics of biological nitrogen fixation. In this lecture, we'll first study briefly about the nitrogen cycle, then we'll study the process of nitrogen fixation by abiotic as well as biological means. Lastly, we'll explore the biochemistry and genetic machinery that derives biological nitrogen fixation in the nitrogen fixing bacteria. The nitrogen is an essential constituent of cell amino acids and nucleic acids. However, the higher organisms are unable to directly utilize the atmospheric nitrogen. This nitrogen is fixed in various forms such as nitrous acid, nitric acid, and ammonia in the soil, from where the higher organisms take up nitrogen for their protein and nucleic acid anabolism. The deficiency of nitrogen manifests in the form of growth inhibition in general.
Nitrogen is present in many forms in the biosphere. For the most part, this large reservoir of nitrogen is not directly available to living organisms. Acquisition of nitrogen from the atmosphere requires the breaking of an exceptionally stable triple covalent bond between two nitrogen atoms to produce ammonia or nitrate. These reactions, known as nitrogen fixation, can be accomplished by both industrial and natural processes. Nitrogen fixation is the process of conversion of atmospheric nitrogen to ammonia. It plays an important role in the mobilization of nitrogen in the ecosystem. Once fixed in ammonium or nitrate, nitrogen enters a biogeochemical cycle, the nitrogen cycle, and passes through several organic or inorganic forms before it eventually returns to molecular nitrogen. This figure shows the nitrogen cycle. The importance of the nitrogen cycle stems from the fact that despite its high concentration in the atmosphere, most of the living organisms are unable to directly utilize free nitrogen for synthesis of nitrogenous biomolecules such as amino acids, purines, pyrimidines, and others. Hence, there is a need to convert atmospheric nitrogen in utilizable form, a process termed as nitrogen fixation. Once fixed in ammonium or nitrate, nitrogen enters the nitrogen cycle and passes through several organic or inorganic forms before it eventually returns to molecular nitrogen. The ammonium and nitrate ions are either generated through fixation or released through decomposition of soil organic matter.
In the nitrogen cycle, there are many related processes such as nitrogen fixation, ammonification, nitrification, and denitrification. Each of them is crucial for the completion of nitrogen cycle. Nitrogen fixation is the conversion of nitrogen into ammonium. So, then what is the ammonification? In fact, ammonification is the process of formation of ammonium ions from organic nitrogen. This happens when the animal or plants die or when animal excretes the waste. Since most plants cannot use nitrogen in the ammonium form, it has to be converted into nitrates and nitrites. This process is called nitrification. The fixed nitrogen from the biosphere is liberated back to the atmosphere by the process of denitrification.
Nitrogen fixation can be accomplished by both industrial and natural processes. The natural processes may be either abiotic, such as nitrogen fixation by lightning, or mediated by microbiota in various ecosystems, such as agricultural land, forests, and marine ecosystems. Industrial processes involved chemical synthesis of ammonia from molecular nitrogen and hydrogen, as well as fossil fuel combustion. Biotic processes in the different ecosystems have a major share in the annual nitrogen fixation, with the agricultural land and forest ecosystem accounting for more than half of the fixed nitrogen. Fertilizer production by Haber-Bosch process is the major abiotic means of nitrogen fixation. The Haber-Bosch process is a starting point for the manufacture of many industrial and agricultural products and involves combining nitrogen with hydrogen at very high temperature and pressure to form ammonia. Natural abiotic processes such as lightning and photochemical reactions in upper atmosphere also have a minor share in nitrogen fixation. Lightning converts water vapor and oxygen into highly reactive free radicals and atoms, resulting in acid rain. Ozone and nitric oxide also react with each other to form nitric acid and acid rain. Biological nitrogen fixation, discovered by Beijerinck in 1901, is carried out by a specialized group of prokaryotes. These organisms utilize the enzyme nitrogenase to catalyze the conversion of atmospheric nitrogen to ammonia and are called as diazotrophs, which are organisms that are able to grow without external sources of fixed nitrogen. The nitrogen-fixing prokaryotes include aquatic organisms such as cyanobacteria, free-living soil bacteria such as azotobacter, bacteria that form associated relationship with plants such as azospirillum. Most important nitrogen-fixing bacteria include rhizobium and bradyrhizobium that form symbiosis with legumes and other plants. Rhizobium is also an important biofertilizer for legume plants. There is no obligatory relationship between rhizobia and legumes, as each is capable of survival without the other. However, under nitrogen-limited conditions, the symbionts seek out one another through an elaborate exchange of chemical signals. The signaling is followed by infection process and the development of nodules. All these processes are driven by underlying genetic mechanisms. The nitrogen-fixing bacteria are endowed with special nitrogen-fixing apparatus such as the NIF genes to encode the nitrogenase enzyme. This nitrogenase enzyme to convert nitrogen to ammonia. There are mechanisms to maintain anoxic conditions such as leg hemoglobin, high respiration levels, or anaerobic respiration, as the nitrogenase enzyme is prone to oxygen inhibition. There are electron acceptors such as ferredoxin or flavodoxins for the reduction reaction of nitrogen to ammonia. Since the process of biological nitrogen fixation is highly energy intensive. There is an abundant supply of ATP molecules. Molybdenum present in nitrogenase and nitrate reductase and iron present in the nitrogenase enzyme structure is obtained from the symbiotic plant partners or the soil. As the presence of nitrate and ammonia in the cell's environment is detrimental to nitrogen fixation, hence these are removed. Root nodules of legumes, Actinorhiza and Gunnera, are the dwelling sites of the symbiotic nitrogen-fixing bacteria such as rhizobium. The formation of these root nodules themselves is induced by the symbiotic bacteria or may be stem glands that develop independently of the symbiotic bacteria. The nodules are pink in color due to the presence of leg hemoglobin, a molecule analogous to the hemoglobin in animals that transports oxygen to respiring tissues in animals. Leg hemoglobin, however, has 10 times more affinity for oxygen than human hemoglobin beta chain. However, the occurrence of root nodules is not mandatory for the purpose of symbiotic nitrogen fixation, as evidenced in some grasses that are known to be symbiotically associated with nitrogen-fixing bacteria that mainly colonize the root surface. Genes specific to nodules in plants are called nodulin, designated with an uppercase N in NOD, whereas genes in the rhizobium are designated nodulation genes and designated with a lowercase n in NOD. The common nod genes are nod A, nod B, and nod C. On the other hand, nod P, nod Q, and nod H, or nod F, nod E, and nod L are host-specific. Nod D gene regulates the function of other nod genes. It is constitutively expressed as the protein nod D. The nod D protein expression is triggered by a chemotactic response in rhizobia. The nod D protein then triggers the expression of other nod genes. These genes are involved in the synthesis of the nod factors. The specific role of these nod genes has been enlisted in this table. The first stage in the formation of the symbiotic relationship between the nitrogen-fixing bacteria and their host is migration of the bacteria towards the roots of the host plant. This migration is a chemotactic response mediated mainly by flavonoids and betaines secreted by the roots. These attractants activate the rhizobial nod D protein, which then induces the expression of other nod genes involved in the synthesis of nod factors. The response of legumes is specific to the nod factor and is mediated by lectin receptors present on root hairs. Nod factors activate these lectins, increasing their hydrolysis of the phospho and hydride bonds of nucleoside di- and triphosphates. This lectin activation directs rhizobia to anchor to the cell walls of the root hair. The stages in the development of the root nodule have been depicted in this figure. Following the chemotactic movement of rhizobia towards the plant cells, infection of the plant cells by the rhizobia occurs in the following stages. In A part of the figure, formation of the infection thread is seen. The nod factors induce a curling of the root hair cells which enclose the rhizobia. The cell wall of the root hair degrades in these regions, allowing the bacterial cells direct access to the plant plasma membrane. The plasma membrane then invaginates and fuses with Golgi-derived membrane vesicles to form an infection thread at the site of infection. In the figure part B, we see the generation of nodule primordial. Deeper into the root cortex, near the xylem, cortical cells dedifferentiate and start dividing, forming a distinct area within the cortex called a nodule primordium, from which the nodule will develop. The nucleoside uridine and ethylene control cell division in the root primordia. Part C here shows the release of bacterial cells into root primordia. The infection thread, filled with proliferating rhizobia, elongates through the root hair and cortical cell layers in the direction of the nodule primordium. When the infection thread reaches specialized cells within the nodule, its tip fuses with the plasma membrane of the host cell, releasing bacterial cells that are packaged in a membrane derived from the host cell plasma membrane. In part D, the branching of the infection thread inside the nodule happens, enabling the bacteria to infect many cells.
First, the bacteria continue to divide, and the surrounding membrane increases in surface area to accommodate the growth by fusing with the smaller vesicles. Soon thereafter, upon an undetermined signal from the plants, the bacteria stop dividing and begin to enlarge and to differentiate into nitrogen-fixing endosymbiotic organelles called bacteroids. The membrane surrounding the bacteroids is called the peribacteroid membrane. The nodule develops a vesicular system which facilitates the exchange of fixed nitrogen produced by the bacteroids for nutrients contributed by the plant, and a layer of cells to exclude oxygen from the root nodule interior.
Steps that were just discussed have been shown in this flow chart here. Bacteroids produce the nitrogenase complex that converts atmospheric nitrogen to ammonium. Without the bacteroids, the plant is unable to utilize nitrogen. The infected root cells provide some factors essential for nitrogen fixation, including leg hemoglobin. This heme protein has a very high binding affinity for oxygen, which strongly inhibits nitrogenase.
In this lecture, so far, we have seen the microbiological aspects of biological nitrogen fixation. Biological nitrogen fixation is a series of enzyme catalyzed biochemical redox reaction. Let's now look at the biochemical aspect of nitrogen fixation. We will look at the enzymatic machinery which is involved in nitrogen fixation, the various factors that control the enzymatic machinery, and other aspects that are critical to the normal functioning of the enzymes.
The basic function of the nitrogenase enzyme is to fix the molecular nitrogen into ammonia, which is usable form of nitrogen for the organisms. It is an intrinsic property of nitrogenase to reduce protons along with nitrogen and to produce hydrogen as an obligatory side product. One molecule of hydrogen and two molecules of ammonia are formed by molybdenum nitrogenase catalyzed reaction at the expense of 16 moles of ATP. This reaction is irreversible and can provide a continuous supply of hydrogen even under an atmosphere of 100% hydrogen. This process consumes large amounts of energy in the form of ATP molecules. ATP is regenerated by photophosphorylation. Hydrogen production on a commercial scale has been done using this enzyme, as in the absence of nitrogen, all the electrons are used in the hydrogen production.
What's unique about the major nitrogen fixation enzyme? The nitrogenase is the primary and the unique enzyme responsible for biological nitrogen fixation. Let's see. It's a tetramer having an iron-sulfur cluster and molybdenum-iron cluster. The enzyme consists of two components. These are molybdenum iron protein known as component 1 and the iron protein known as component 2. Well, we'll study more about this enzyme later. Let's again understand here that this key enzyme catalyzes the conversion of dinitrogen to ammonia. In the biological nitrogen fixation reaction, dinitrogen reacts with water and ATP in presence of electrons to form ammonia, dihydrogen and protons, and liberation of ADP and inorganic phosphate follows. In this reaction, dinitrogen is captured from atmosphere, whereas the hydrogen atoms are donated by water. The process is quite energy intensive, hence the requirement for large number of ATP molecules.
The reduction of dinitrogen to ammonia is coupled to the reduction of two protons to evolve dihydrogen. This reaction is catalyzed by the nitrogenase enzyme complex. Nitrogenase is an alpha 2 beta 2 tetramer. The alpha and beta subunits have a similar size and are similarly folded. The tetramer contains two catalytic centers, probably reacting independently of each other, and each contains a so-called P cluster consisting of two 4-iron 4-sulfur clusters and an iron molybdenum cofactor. Iron molybdenum cofactor is a large redox center made up of iron 4 sulfur 3 and iron 3 molybdenum sulfur 3, which are linked to each other via 3 inorganic sulfide bridges. The enzyme consists of the molybdenum iron protein known as component 1 and the iron protein known as component 2. The molybdenum iron protein has 4 subunits with a total molecular mass of 180 to 235 kilodaltons depending on the species. This subunit has the dinitrogenase activity. The iron protein has the dinitrogen reductase activity and is the smaller of the two components and has two identical subunits of 30 to 72 kilodaltons each, depending on the organism. Each subunit has two molybdenum iron sulfur clusters. Both the subunits are required for the reduction of dinitrogen to ammonia and are susceptible to irreversible inactivation by oxygen. For activation, dinitrogen reductase requires ATP, which gets hydrolyzed to ADP and inorganic phosphate. Only such activated dinitrogenase can mediate the transfer of one electron at a time from reduced ferredoxin or flavodoxin to dinitrogenase, which contains the sites for its substrate. Such repeated one-electron transfer processes result in the building up of a pool of electrons in dinitrogenase, which are finally used to reduce nitrogenase substrates. In the overall nitrogen reduction reaction, ferredoxin serves as an electron donor to the iron protein, which in turn hydrolyzes ATP and reduces the molybdenum iron protein. The molybdenum iron protein then can reduce numerous substrates such as hydrogen cyanide, azide, or acetylene, although under natural conditions it reacts only with dinitrogen and proton. There are numerous factors that influence the activity of the nitrogenase enzyme. These factors are depicted in this figure. First is the availability of molybdenum in the soil, as it is necessary for the formation of the nitrogenase enzyme. Secondly, as the hydrogen produced during the process of nitrogen fixation has an inhibitory effect on nitrogenase, the membrane-bound enzyme uptake hydrogenase is used by the nitrogen-fixing organism to quench the hydrogen produced during nitrogenase activity. Thirdly, as already mentioned, oxygen is a strong inhibitor of the nitrogenase activity. The problem of oxygen is countered by various mechanisms discussed in the next slide. The problem of oxygen inhibition of nitrogenase enzyme is a crucial one, and various mechanisms are adopted to overcome this problem. These include anaerobic respiration, modified respiration rate, regulation of nitrogenase gene expression, and the presence of specially modified cells known as heterocysts in the nitrogen-fixing species. Leg hemoglobin is yet another mechanism of preventing the damage due to oxygen. Let us look at each of these mechanisms in more detail. To solve the oxygen toxicity problem, the bacteria in root nodules are bathed in a solution of the oxygen-binding heme protein, leg hemoglobin, produced by the plant for which the heme may be contributed by the bacteria. Leg hemoglobin binds all available oxygen so that oxygen cannot interfere with nitrogen fixation, efficiently delivers the oxygen to the bacterial electron transfer system. Leg hemoglobin, formed in rhizobium, acts as the reservoir of oxygen for the bacteroid for aerobic respiration, as well as to bind and quench the diffusing oxygen within the nodule. In the part A of the figure shown in the slide, you can see the artificially colored electron micrograph of a thin section through a pea root nodule. Symbiotic nitrogen-fixing bacteria or bacteroids are seen as red structures inside the nodule cells, surrounded by the peribacterioid membrane, which is blue. The heterocysts are specialized nitrogen-fixing cells of filamentous cyanobacteria such as nostoc and Anaerobic. They are exclusive sites of nitrogen fixation. They fix nitrogen using the nitrogenase enzyme. They create a micro-anaerobic environment for dealing with the oxygen problem by various mechanisms, such as by synthesizing 3 additional cell walls. One of these cell walls is a glycolipid cell wall. These cell walls act as a hydrophobic barrier to oxygen. Secondly, the photosystem II, which is responsible for the liberation of oxygen, is absent in them. The heterocyst cells selectively degrade their photosystem II by means of specific proteases. Further, the glycolytic enzymes are also overexpressed in the heterocyst cells. This increases the rate of glycolysis to meet the increased energy demand of nitrogen fixation. Cyanobacteria, like nostoc, use oxygen scavenging proteins such as cyanoglobin for oxygen scavenging and thus creating an anaerobic microenvironment for nitrogen fixation to occur. This protein is specifically localized to the peripheral membrane of the heterocyst. One in about 9 to 15 cells in the cyanobacteria filament develops into a heterocyst. In the later stages of the heterocyst development, there is accumulation of the polypeptide known as cyanophysin. This is important for the storage of the fixed nitrogen in the heterocyst, as well as to prevent the diffusion of oxygen from the adjacent vegetative cells.
Klebsiella pneumoniae overcomes the oxygen problem by repressing the production of nitrogenase enzyme in presence of oxygen. Since efficient operation of nitrogenase requires considerable amount of ATP and reducing power, synthesis and activity of this enzyme in response to environmental stimuli are subject to strict regulatory controls at transcriptional and post-translational levels. In photosynthetic bacteria, the physiological regulation of nitrogenase activity involves inactivation by covalent modification of the iron protein of nitrogenase. This inactivation can be reversed by an activating factor, which is extrinsic membrane protein. The primary inhibitor or repressor of nitrogenase is oxygen, which interferes with biological nitrogen fixation at different levels. At genetic level, oxygen represses the nitrogenase synthesis by downregulation of the expression of NIF genes. Furthermore, due to its oxidative properties, oxygen causes irreversible damage to the enzyme by degrading the iron-sulfur cofactors. Dinitrogenase reductase, the iron protein, is more sensitive to inactivation by oxygen than dinitrogenase, the molybdenum iron protein. The nitrogenase activity of the cells might be restored partially or completely by re-establishing anaerobic conditions after respiring the traces of oxygen present in the culture. Furthermore, once the anaerobic conditions are set, new nitrogenase enzymes will be synthesized. Ammonium is another inhibitor of nitrogenase. It represses both synthesis and activity of nitrogenase enzyme. nitrogenase B and C and glucan BK are the two component system which sense the fixed nitrogen status of the cell and accordingly regulate the NIF operon and NIF-A gene. The gene product of NIF-A, which is a member of enhancer binding proteins, is a transcriptional activator of NIF structural genes in bacteria and thereby regulates the activity of NIF genes. The presence of ammonium ion in the culture medium causes a decrease in the expression level of NIF operon and NIF-A gene and results in decrease in hydrogen production. At the post-transcriptional level, the nitrogenase enzyme activity is reversibly inactivated in a way that an ADP ribose group from NAD is attached to an arginine residue in one subunit of the NIF-H protein, resulting in NIF-H inactivation, which is called switch off. This process is catalyzed by nitrogenase reductase or ADP ribosyl transferase called DRAT. However, this inactivation is reversible such that when the added ammonium is exhausted by cellular metabolism, the ADP ribose group is removed by dinitrogenase reductase activating glycohydrolase DRAG, leading to NIF-H inactivation, what is called as switch on. However, nitrogenase biosynthesis is strongly stimulated by light. It was observed that nitrogenase represent up to 40% of the cytoplasmic protein in RB. Capsulatus under high illuminations. It is also worth noting that a diurnal pattern of illumination, which means one involving alternating periods of light and dark, rather than continuous light, results in a more stable nitrogenase activity. Free-living nitrogen-fixing bacteria are both aerobic, facultative, or anaerobic. Anaerobic nitrogen-fixing bacteria may be either facultative anaerobes or obligate anaerobes. Facultative organisms which are able to grow under both aerobic and anaerobic conditions generally fix nitrogen only under anaerobic conditions. The anaerobic nitrogen fixers take care of the oxygen problem by their very anaerobic mode of nutrition. For them, oxygen does not pose a problem because it is absent in their habitat. These anaerobic organisms can either be photosynthetic, rhodospirillum for example, or non-photosynthetic Clostridium for example. Anaerobic respiration is a form of respiration using electron acceptors other than oxygen. These include sulfate, nitrate, sulfur, or fumarate. These terminal electron acceptors have smaller reduction potentials than oxygen, which means that less energy is released per oxidized molecule. Anaerobic respiration therefore does not pose any problem of oxygen inhibition to the nitrogenase enzyme in general, though energetically it is less efficient than aerobic respiration. These nitrogen fixing species include Clostridium acetobutylicum, Clostridia bisrenkai, Rhodospirillum rubrum, Enterobacter agglomerans, and Enterobacter oryzae. The aerobic bacteria such as Azotobacter protect their nitrogenase by adjusting the rate of aerobic respiration such that an aerobic environment is created within the cell engaged in nitrogen fixation. To summarize the nitrogen cycle, this cycle is responsible for mobilizing the atmospheric nitrogen into soluble form by the process called nitrogen fixation. This nitrogen fixation may occur both abiotic and biotic means. Biological nitrogen fixation by the rhizobium legume system is by far the most important type of nitrogen fixation. Genetic and biochemical basis of biological nitrogen fixation, and lastly about the factors that affect nitrogenase enzyme function have been studied in detail. So students, in this lecture, so far we have learnt about the nitrogen cycle in the environment, abiotic and biotic modes of nitrogen fixation, biological nitrogen fixation in rhizobium legume system, genetic and biochemical basis of biological nitrogen fixation, and lastly about the factors that affect nitrogenase enzyme function.